Ready to get here. Jesus Christ, I get to meet you guys. All right, hey, what's up? You guys can sit where you like. But you can't, but you got to stick there. Understand when you make that decision, it's a hard, she just sat there and did not get a chance to clean hers. I wouldn't choose that one. I choose this one before I turn it that way, unless you want to clean it. You want to wipe it down? I can wipe it down for you. You want me to wipe it down?
I'll do it.
You don't want? I'd love to serve you like that. Can I do that? Can I be nice like that? You don't want that little kids around. It's terrible. What's up, kiddo? Say, holy smoke, man! I finally get to put eyes. I get to put eyes on your one eye. All right, you guys come on in. Huh? Nobody's up there. Oh, come on. Could you walk up here, please? Please.
You look like a little kid.
Good morning, beautiful people.
Good morning.
We're running a little behind here at the school today. These poor children didn't get a chance to eat anything. So sorry about that. Make sure I get to your class. This is 3A. Excellent. That is the wrong date. Class content.
Every time you leave, you take the pictures with you.
No, they have to struggle bus it. What's up, boys? Did you know that new sub here, her name's Boys too? The lady sitting on in the hallway. She's you'll see her. She's she took my name. No, I think she's older than you. You took hers. For you guys at home, the students, some of the students that are just getting in the class, lunch was about 20 minutes delayed today. So there's a bunch of kids. I can't hear. What?
I'm talking to Lazarian who had a hood on her head.
Oh.
We don't allow them.
So we'll start at, say, 12:05 to let everybody get in here. There's still people literally coming in the room, guys. You got to give them a little bit of a break. Sure, absolutely. Mi casa, su casa.
I go to the bathroom real quick.
Yeah, do it. Well, actually, we have to wait until class starts. I can't let people between classes. So as soon as I get started, or let, whoa, hold on a second. Where are you getting? Boyce just asked he would go first. So what you need to do is there's a place to sign in and sign out here, and then one at a time. But Boyce had already asked, dude. Sorry. As soon as he gets back, do it. All right. So again, I'm sorry that it's taking a little longer. Everybody's still getting in here. Get started. We'll start at 12:05. In the meantime, please go to the class website and go to Cell Cycle. January the 6th, Cell Cycle. That's where we're going to be working. While we're waiting, anybody have any exciting news? Something happened? I had a couple of you guys ask how my break was. My break was fine. I sent you guys pretty pictures. I got to be out in the snow. I hope a lot of you guys got to see the snow. If you didn't get a chance to see the snow, you really didn't miss a lot because it only stayed around for about 7 or 8 hours and it was all gone. You didn't miss much at all.
Wait, where was the snow?
It snowed about 100 miles west of here. There was a lot of snow. About 100 miles west and northwest. If you went southwest, it really started to peter out pretty quickly. Looks like we still have people getting it. But it was beautiful. I mean, it was beautiful. You guys should have seen the photos I sent on Remind. I sent a couple of really pretty pictures. I mean, it was a lot prettier in person. You guys also, while we're waiting another 2 minutes, had asked that we would be able to have some practice, like multiple choice and open answer, open response practice without there being grades attached to it. So we're going to do some of that today as well. In first period, I got a new account for Zoom and I had no breakout rooms, and now I do. So we'll be able to do this whole lesson, no problem. Okay, well, while we're waiting, you guys, cell cycle is the topic. When you're looking at this image, I've got a GIF here, a little GIF, whatever you call those things. If you, in one word, could describe this, describe it in a single word, and please put it in the chat. And leave it open to everybody. I want to see what kind of adjectives or verbs we could get for this. Try to make it a verb or an adjective, maybe. What do you see occurring? What do you see occurring? What do you got?
DNA being multiplied.
Oh, do it in one word. If you could just put it in a word, a single word, what word would you use? What verb? You try to use verbs or adjectives.
Divide.
Okay, divide, division, replication, divided, division, a lot of, okay, keep it up. Duplicate, nice. Good, duplication, Lazy Aguilar. Process, separate, split. Like a banana split at Lowe'sman? Banana splits are delicious. No, I don't like banana splits, to be honest with you. Generate, Jalen gives me generate. Detach, okay, detach cycle. You guys are a lot more creative than my first period. I think they came up with 4 different terms and that was it. Undertaking. Oh, that's where you take a person and embalm them before they die, right? There's Monk. Okay, it's 12:05, and I'd like to start with the things that you guys put in the chat. We are going to be dealing with the topic of the cell cycle. Our new unit is going to be genetics, so we're going to just slowly ease into genetics. I need somebody to set a timer for 15 minutes and then come on the microphone and tell me shut up. And people in this room can all yell shut up. If Safe yells to shut up, I'd be even more impressed. But I'd love it if you guys would just stop me within 15 minutes. Man, hush, I'm tired of listening to you. Shut your mouth. So you guys use the term, a lot of terms. You said division, multiplication, separating, dividing. You guys said a lot of things. And I really wanted to get into that because a cell cycle is really centered on 2 big principles, and that's growth and division. Remember that cells cannot, thank you, cells cannot grow large. If they do, their surface area to volume ratios decrease, and if they decrease, diffusion stops or slows, and they do not want to lose that process. It's a free delivery service, you can imagine, as a delivery service, and so if you lose that delivery service, it's going to cost you more energy. You have to go active transport. So if you're going to grow and you're a cell, the only way to do it is to divide, is to grow in number, not grow in size. Now, not all cells do this at an even pace, and you can imagine why. So the first scenario I have here, it says we're using fly embryos. So the cell cycle in a fly embryo is 8 minutes. So imagine you start with 1 cell, a fertilized egg, an embryo, and you go in 8 minutes, you go from 1 to 2, and then 8 minutes later, 2 to 4, and then 4 to 8, 8 to 16, 16 to 32, 64 to 128, and you just get more and more and more. It goes really, really quick. So within a matter of literally hours, you go from a non-visible to the naked eye cell to a visible maggot or a larva. They grow very quickly. Now, why is that? Why do you imagine that a larva or an embryo, or even you could even consider a child, if you want to make an analogy for a person, why do you think children grow more quickly? Why do young organisms grow fast?
Your question was, why do non-organisms grow fast?
Why do young organisms?
Oh,
Imagine a baby, or a baby, a puppy, a kitten, a little infant bird, a chick, a pup. Why would you grow fast? What would be why would you need to do that?
so they can survive on their own.
Okay, but okay, absolutely. They want to grow up to be able to survive on their own, but you can get that training a little later at a bigger size. Why get big quick? What is that going to decrease? And let me give you also another fact. I'm going to drop facts here. We're not going to go on opinions. No conspiracies. 99% of everything that's ever born dies before it reproduces. The exception is our humans, which it used to not be an exception, but it is now because we've got modern technology. Why? What do you think happens to that 99% that don't survive to adulthood? And I know die, but why did they die? What's the biggest reason? Throw it out there. What do you think? I know you guys are city kids, but you probably have still got some clues, man. What happened? There's yeah, they get eaten. Thank you. There were Abby put in there that they get eaten. That's right, predation. So your job as a little bitty organism is to get big fast so I don't get eaten. That's a big motivator, right? So that is going to be important. Now, that's some cells, though, when we start thinking about organs, don't grow fast. The cell cycle takes a long time. For a liver, it takes up to a year. So your liver as an adult doesn't grow very quickly, doesn't repair itself very quickly. So if you damage your liver, you're done. So this always, it really does worry me because your liver detoxifies your body. If you eat anything that is not a normal substance, even considering like food color and other items, those things have to be detoxified by your liver. Your liver, as it dies, if its cells are damaged, they can't be replaced very quickly. So if you do, especially using drugs or alcohol, you damage your liver every time you do this, it can't repair itself in your lifetime. You shorten your life. And so your liver is something that you got to understand is going to grow very quickly. An organ that would, what do you think a cell cycle on an organ in your body that would grow quickly, that would go through the cell cycle quickly? Any ideas? Quickly, like some organ that you know would have to be replaced often.
Skin.
Oh my God, yeah, skin. People are like, oh my God, I got some ashy skin and crap, right? So like your skin is constantly dying and constantly being replaced. So I'm looking at Cam. Cam's sitting here with his beautiful head. That head that I'm looking at on Cam, that skin on Cam, that skin that I saw in August is different than the skin I'm looking at now. Nothing I'm looking at I've ever seen on Cam in August. All of it's new. What, you think that's crazy? There's all these kids looking at me like that's crazy. It's true, man. The mother you saw last year is not the mother you saw this year. I mean, it's the same person, but the skin's new. And so, yeah, skin's going to replace itself quickly. All right, I've spent a little too much time. All right, so there's different parts of the cell cycle. We're going to spend a great deal of time on this part, the interphase, on Friday. I'm going to barely go over it now. Interphase, you can see, is most of the cell cycle. It's not a lot going on during that phase. Actually, there is, but it's more subtle. There's G1, S, and G2. But you'll notice there's two main phases in the cell cycle. There's going to be the meiotic phase here, very short little arrow. And then you have interphases where much of the cell cycle is taking place. Interphase just really means between phases. That's really what it means. It means between. That's what inter means. So it's between phases. There's three parts, and you really don't need to know a whole lot about it today. You need to know more about it on Friday. So I'm going to really gloss over this, and don't worry. There's G1. That's protein synthesis. G1 is where the cell is actually carrying out its normal function. And there's a little caveat to that. We'll deal with that on Friday, but that's where the cell is actually doing its job. It's secreting, it's metabolizing, it's doing whatever it does. And then the synthesis phase is where you copy your DNA. And that's going to get prepared to go into mitosis, but the cell still has a normal function, but it's copying its DNA. And this would be an example. So I go from one chromosome to doubling those chromosomes. The chromosomes are connected by a little button-like structure. I always think about it like a button, because a button keeps your shirt together. A button keeps these, what we call sister chromatids together. This is a chromosome, but a replicated chromosome is going to be called a sister chromatid. We name things most of the time with a female gender. So like people usually name the car, they go, this is her, right? Well, we also will name these chromosomes with the female prefix, so to speak. It's a sister chromatid. G2 organelles double. So if you're going to divide, you need more mitochondria, more endoplasmic reticulum. You need more of everything that you're going to need for that cell to function. So your organelles divide, and then you go into mitosis. I'm going to deal with this a lot more on Friday. What you need to worry about today is just what it looks like. So when you look at this cell, it looks pretty normal, right? You get a cell membrane, but this is where we want to pay attention to is the nucleus. The nucleus has a membrane all the way around it. We call it the nuclear envelope. And you have these two little structures called centrioles, and you'll see them in a second, what they do. But there's not much happening. It's just normal. And I'll come back to it again in just a second. So what we do need to remember, again, this is what it's all about, is mitosis is basically about growth, and growth by dividing. But it could also be asexual reproduction. If you're a unicellular, a single cellular eukaryote, only a eukaryote, it would be a single cell. A single cell would need to divide and to grow, because they don't have sex organs. If you can find a cell that has a penis or a vagina, I would really be interested in seeing that, because those are organs, and organs need to be made of tissues, and tissues made of cells. If you don't have organs, the only way to divide, the only way to reproduce is to go through asexual or non-sexual reproduction. So it could be used for that as well. But mitosis is about dividing this nucleus. And so let's get from interphase into something called prophase. So you're going to produce identical daughter cells, and you want them to be identical, but it's going to be you. You want to produce another copy of you. Cam wants to produce Cam, Pope wants to produce Pope. Everybody wants to produce another copy of themselves. You don't want another person growing out of you, another organism growing out of you. There's an acronym that's easy. You probably remember this from your freshman years. PMAT, prophase, metaphase, anaphase, telophase. Just PMAT is an easy way to remember this. You'll get a chance to practice it in about 10 minutes. But that's just the order. Let me show you what characteristics they have. Apoptosis, I'm going to deal with this more on Friday as well, but you do want your cells to sometimes kill themselves, especially if there's an error, and we'll come back to this, but apoptosis really means cell death. And again, Friday, I'm going to deal with this, not today. Let's do a little math. So there's a term called ploidy, haploid, diploid, and diploid is 2n. That's the symbol for diploid, 2n. Now, this is just simple multiplication. If n equals 23, what does 2n equal? So what is 2 times 23? Don't answer out loud. I'm going to ask one of you weird kids. Let's go to Stein and my eye. If n equals 23, what does 2n equal? What is 2 times n? You can drop it in the chat or the microphone. I don't really mind either way.
Yes, Coach Lee.
Yeah, Stein, what is 2n? What is 2n equal to, kiddo? What is 2 times 23? Correct. Oh, wrong person. I'm asking Stein, man. Why did you, oh, there she is. She did it. Thank you. Yeah. It's going to be 46. So 46 is our diploid number, and you do need to know what that number is. Our diploid number is 46. All of us have 46 chromosomes. Obviously, I have to replicate those to divide. So we go from 46 to 92 and then split into 2 new cells. It's not that difficult. All right, let's look at characteristics. This is part. You want to write this down as I do this. You want to take some notes. Good job, audio. So first phase is prophase. Pro is before, really means before. So it's a before phase, before dividing. And you'll notice that there's these structures called spindles. There's centriole, and there's these little fibers called spindles. There's something else is different about this nucleus. The nucleus, the envelope is breaking down, it's degrading. And so you'll see the centrioles start to move to opposite poles. We call it, like, think about a cell having like a north pole and a south pole, opposite ends. And the chromosomes become visible because the nuclear membrane is disappearing. It's breaking down. There's a set of enzymes that start to chew this membrane up and break it down so that you can, you know, if you take something out, these are big objects. They can't diffuse through a membrane. So they have to be broken down. And I've given you guys a copy of this as well. You're going to have to come back and use this PowerPoint in a moment to do your group work. Metaphase is a little different. Metaphase means middle phase. And it's easy to remember because M is meta and middle starts with M as well. Now, if these are the poles, then the middle of the cell is going to be the equator. Think of it like a globe. So you'll notice the centrioles are on opposite poles. Whoa, I didn't mean to do that. They're on opposite poles at this point. But the main character that you really want to remember is like these sister chromatids, these chromosomes, they're lined up. These spindle fibers are attached to that little button, that centromere, and they'll pull these chromosomes apart. And that's the metaphase. It literally looks like this. It's super easy to recognize. You'll get a chance to practice in a little bit. Anaphase is away, not fully away. They're still near each other, and they're getting pulled to opposite poles, but the sister chromatids have been torn apart. They've been broken. That centromere is degraded, and they've been pulled apart. So there'll be 46 chromosomes going to the South Pole and 46 chromosomes going to the North Pole. They're going to see Santa Claus, and these are going to see the penguins. That's just me trying to be funny. It's not true. Yeah, Santa Claus is not in your cells. Oh, by the way, I did have a kid about 10 years ago that was in high school that still believed in Santa Claus. I had no hope for that child. But the centromeres are going to, they're going to divide. The chromatids are going to separate. You're going to opposite poles, opposite sides. It's really easy. It's the away phase. But to go far away, if let's say Daniel, if you wanted to talk to me, I'm at home, what would you do? You wanted to talk to me at home, what would you do? Daniel's in the class with me. You want to talk to me, what would you do? I'm at my house, you're at your house. Exactly. You call me. Exactly. So the last phase is the far away phase, and think of it like a telephone. When you know that you'll notice that those chromosomes are no longer being pulled by spindle fibers, they're gone. The nuclear envelope is going to make these sister chromatids be far away. Those chromosomes start to get grouped together so that they can be in different cells. They're far away from one another now. They're not close to one another. They're not attached to spindle fibers anymore. You have this little furrow. This is called a cleavage furrow, like a pinched end. If you have a meat cleaver, you're cutting meat apart. You cleave the meat. You cut the meat. That's what cleavage means. If you're a young lady, your mother may say, hey, man, you want to show your cleavage. That's that little pinching in between your breasts that you want to pull up your shirt so that's not exposed. That's what your parents have probably told you. And so chromosomes are going to also unwind. Is that 15 minutes?
Yeah.
Perfect. Y'all going to say anything? What are y'all going to say?
I was waiting for you to finish talking. Don't want to interrupt you.
Well, I mean, that's what I told y'all. What y'all going to tell me?
Shut up.
Oh, man, that's what somebody shut up. Come on, man. Y'all are scared. Don't be scared.
Mr. Poe, question. How
What?
is the slide in class, like you can find it?
Yeah, it's under today's lesson. It's 1-6. Yeah, this whole PowerPoint, huh?
Okay.
Oh, yeah, knock yourself out. Okay, so that was my 15 minutes. All right, get ready. Actually, can I have 90 more seconds? So, okay, thank you. Javier says sure. He said I could have 90 more seconds. So I got to show you one more thing. Cytokinesis. Cytokinesis is the very last step. Cyto means, what does cyto mean again? C-Y-T-O? Cyto. How y'all think? What do y'all think? Come on. What are we talking about? What is this thing that we're looking at?
A cell.
A cell, exactly. Thank you. Cell. In kinesis, if you're a kinesiology major, what do you study? A kinesis. Movement, that's right. Movement is kinesis, right? If you're a kinesiology major, you study sports. People move with sports. That's why video games is not a sport. Esports is ridiculous. There's no movement except for your thumb. Silly. So cell movement, at this point, cells are moving apart. They're separated, essentially. They have actually separated. So there's one thing that can distinguish animal cells from plant cells, and this is essentially it. You can see what we call is a cell plate, and this is a cell plate that will become a cell wall. That's the biggie. That's the big difference. There's no cell walls in animals, otherwise it would be rigid. But plants are going to have also form a cell wall. You'll notice there's no cleavage furrow. These pinch inward and split. These don't pinch in, but they can't. They're going to be having a cell wall, and then that membrane will form inside of that new cell wall. That's the big difference. Okay, so let's do some practice. All right, I've got some cells here. I'm going to point to a cell, and I only want you to look at it. And then I'm going to point to a cell, and then one of you guys is going to have 30 seconds to, no, not 30. We don't have that much time. Let's take 15 seconds, and you're going to tell me, is it interphase, prophase, metaphase, anaphase, telophase, or cytokinesis? And if you do need today's lesson, you can go to the website and pull up this PowerPoint yourself. It's under today, January 6th, and it says cell cycle. It's pretty straightforward. I'll give you guys 15 seconds to look at these and start really thinking about what you would say if I were to call on you. And I'll call on a lot of you. I've got about 30 of these to go through. So probably all of you will get one. I'm going to go with my favorite student first. In that case, my favorite student, that's going to be, no, that's not my favorite. Hold on, let me think about this. Who's really my favorite? Let's go with Emma. Emma, tell me, what do you think, which phase this cell is in? This cell right here. Yeah, why don't you come on the mic? If you yell enough, they can hear you through my mic, probably.
Metaphase.
Metaphase. How do you know? No, yeah, how do you know? You're right. How do you know that?
Because it's in the middle.
What's in the middle? You said it.
The chromosome.
Yeah, exactly. The sister chromatids. I would describe them as sister chromatids, but the chromosomes are in there. Look at you, man. I'm happy. Okay, cool. Pass it to somebody.
Tabitha.
Hurry. Tabitha. What about this one? Look at that cell. That's a good looking cell.
That one?
See, this one.
Okay. I'm gonna guess here and say
That's a
it's
tough
an
one.
interphase.
I can see your argument there, but I really don't think so.
No.
The reason why is I think it's almost interphase, but it looks like it's kind of going into prophase. You can see like a darker region here, and that might be where the chromosome, and you could probably argue that. If this was open response, you might be able to argue it. But there's some condensation of chromosomes, I'm thinking here. Like these little lighter areas is where the envelope, the membrane, the nuclear membrane or nuclear envelope has actually started to break down. I would err
So
on
basically,
prophase.
this one's prophase because it has the black lines, which are the chromosomes.
Yeah, I think they're starting to condense. You can make that argument. They're starting to condense. Like this one, that's clearly interphase. See how there's not any really darker region? Maybe here there is. So I would argue this is prophase, and that's probably interphase.
Okay, that makes sense. Thank you.
Excellent, excellent. Pass it to the next person.
Uriel.
Uriel Chavez. What about this cell right here, baby? Chavez. I think it's Chavez, right? Uriel, you heard her calling you, right, baby? Is he here?
Oh, sorry. Yeah, I just chat you that I'm away from the keyboard. My mom needs to speak with me real quick. just really quick. Sorry about that.
Okay, cool. I'm gonna jump over here to Mukhtar. Mukhtar, what about this one? Don't tell me your mom's trying to talk to you. Your dog's eating your homework either. What do you got, Mukhtar? Help me out. I want to get you guys to the guy to practice a little more practice. Ooh, Mukhtar's gonna type to you. Nice, Mukhtar. Dang. Nope, that wasn't her. Mukhtar, this cell right here, yo. You want to talk to somebody, you can phone a friend. You want to phone a friend or get some help? Anaphase, you are correct, ma'am. You are correct, ma'am. You can put it out there, everyone, and you can see the sister chromatids have been pulled apart. You can see it. There's probably a centriole here and a centriole here. The spindle fibers have got these sister chromatids centromeres, and they're pulling them apart. Coolio. I'll call on somebody for the sake of time. So let's go to Romello. Romello, what about this cell right? Good grief. What about this cell right here? Salinas. Y'all don't know I went to Salinas' house over Christmas.
Wait, can you say that again, sir?
What?
Can you say that again?
Yeah, what phase would this cell be in?
Would it be S2?
No, S2. S2, you really wouldn't be able to characterize it. It's just going to be like interphase. G1S and G2 are going to be, you wouldn't be able to notice just by looking at them.
Huh. I need help on this one, sir.
Okay, cool. Let's go back and look. So you tell me what it would look like. This, prophase, or would it be interphase? There, right there. There's this image. That's interphase. This is prophase, metaphase, anaphase, telophase, cytokinesis. So, what do you think it is?
I think it's either metaphase or anaphase.
Metaphase or anaphase? Not it. Anybody want to help him? I'm sorry? Telophase or I say telo, but telophase is totally fine. I mean, it's probably what most people say anyway. I'm just kind of a weirdo. Why do you say that, Lazaria? See, this one that I'm looking, yeah, right here. Why did you say that? Yeah, it's dividing. You can see there's probably the cytoplasm still connected right here. They're not completely separated. I think that would be telophase. All right, so let's go to the next contestant on the Pope is right. Taylor Tubbs. What about this one, Taylor?
Can you hear me?
Yeah, I can hear you.
Which one? I didn't see the mouse.
Right there.
Anaphase.
Anaphase, absolutely. They're being pulled apart. Centromeres on opposite poles. The sister chromatids are being torn apart. They're still connected here, probably somewhere between metaphase and anaphase. It's leaving metaphase and going into anaphase clearly. Okay, let's just do rapid time. I'm gonna call on you. You got 5 seconds. So let's see. Cam, this one. Anaphase is correct. Yes, sir. Boom! I love it. Brandon. Donde? Okay. Chromatids are at the equator. I says the sister chromatids are at the equator. The centrioles are at the poles. The spindle fibers are connected. What phase is this? Metaphase. Okay, nice. Mariella. Nuclear envelope is broken down. Centrioles haven't moved to the poles yet. And they're starting to. They're not at the poles, but they've started to move. Not quite. No, the spindle fibers haven't grabbed the sister chromatids yet. Metaphase is right here. That would be their line up at the equator. That's metaphase.
Oh, yeah.
Sorry? Prophase it is. Prophase it is. Nice. Coolio. Let's go to the most wonderful child in the room. That's going to be Jalen Clark. Come on, Clark. What phase of the cell cycle is this cell in, man? It's the same question.
Interphase?
Interphase is correct, sir. Yes, sir. This is normal. There's not even chromosomes. It's just chromatin. It's just loose DNA. Excellent. Nice one. This would be Alosa.
Telophase.
Telophase. Yes. Boom. Let's see. Earl. Eral.
Anaphase.
Anaphase is correct, sir. Excellent. Abigail.
Would that
Abby.
still be interface?
Not quite. This is going to be prophase, and I'm going to show you why on the next slide. This, you can see, just like for Tabitha, you can see where the envelope is being broken down, the nuclear envelope, and the DNA is starting to condense. That would be interphase. Just
Mr. Pope, I have a question. Oh.
I believe you. Go for it.
For the one on page 18,
Oh God.
Is that one? What did you say that one was?
Page 18. Which one? Tell me when I get there, because I can't see the page number.
That one. The one.
This one? This one?
Yeah. No. The one before that.
This one?
That.
That would be interphase, and it looks.
The one before that.
This one?
That has like lines. Yeah, that one.
Yeah. So the lines are the spindle fibers. The spindle fibers are forming between these centrioles. It's kind of a cartoon version of it.
Oh, and it's which one?
Prophase.
Okay.
Excellent. All right, so here we go. We're going to start practicing. Everybody should be going to the website, going to our class. Make sure that you guys can see this. I'm going to give you directions. You're going to have 15 minutes. Now, you guys asked that I give you guys some time to practice without grades, and I'm going to, plenty of time. Today, you're going to get 15 minutes just to practice, and you're going to get to collaborate. This is the only thing I'm taking a grade for today. You'll have a quiz next time. But I'm going to do a little practice with multiple choice before we leave. And multiple choice that's going to be similar to what you'll be doing on Friday. But before we do that, we need to practice this a little more, let you guys talk to one another. So everybody should open this document, cell cycle ordering. Go ahead and click on that and open that. And I'll go through here and I can see who's opened it. So like Daisy, I need you to get yours. Precious, I need you to get yours. Cam's on his way. Uriel, Jalen, Losa. See who the first person is going to be. And I'll take attendance by those who've opened this document. Reggie's first. Yay, Reggie. I'll take attendance by whoever has actually opened this document and completed it. You don't have to complete it till midnight, but, well, I'm going to open it and I'm going to show you what it looks like so that we can kind of work on this together. I'm going to open the document completely. Can you guys see the document? I want to make sure that you guys can see it.
Yes, sir.
All right, excellent. So I have these out of order. They're not in the proper order. Now you're going to have interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So you're going to have to put them in the correct order. The right side, this side, has them out of order. You need to take it by click on the image, right click, cut, and then drop it in the correct order. So you need to order them. But in order, so this side is going to be blank when you're done with it. There will be nothing over here. This side is going to be filled in. These images on the right are going to be on the left side in the correct order. And you're going to be able to, you have to give me 2 reasons you said this, 2 characteristics. What is some evidence in this image? And you can go back and look at the PowerPoint and what is some evidence in the PowerPoint that you can see that justifies, and use the points that I put in that PowerPoint, because those are the arguments that you'll have to make to score points on open response. Why would you say that that image would be in that location? So if whatever goes here has to be interface, whatever goes here would have to be proface. Start with interface. And then give me 2 pieces of evidence. So questions before you guys begin. I'm going to put you guys in breakout rooms and let you guys work on this together. Yeah, this will
What
be
did
your
you
first.
say?
This will be your first grade. Jalen asked me that. What? Go ahead, Elosa.
For the images in order of the, do we rearrange the?
You're
Can
going
you
to
explain
rearrange
that part again?
Yeah, you're going to rearrange these images on the right. You're going to put them over here on the left in the correct order. They're out of order over here. They're not in the correct order. They're going to have to be put in the correct order over on the left side. Any other questions? Okie dokie. You should be able to share your screen. I'm going to make sure that you guys can share your screen. Let's see. You can now. Let's make sure. You don't really need to share your screen, I guess, but if you want to share your screen, you're able to. I'm going to put you in groups of 5 to 6 people. You should be able to open your rooms now. Got 15 minutes, kids. Excellent. Excellent. What? Excellent. You're opening that. Opening that. Okay, let's go. Yeah, hold on. You guys got to swap away. Talk to each other. That's fancy stuff. Hey, what's up? Just see if you guys need anything. Oh, please don't be quiet. You guys got to talk to each other.
I'm doing fine. But do you want us to label the phase after we look the pictures, or should we just make you assume?
Do what again? Do I need you to label the structures? Is that what you're?
No,
So
no,
what
no,
you-
no. I mean like the steps. So like whenever we move the pictures in the correct order, do you want us to like write like interphase and prophase?
You don't have to, but you could. I mean, if it helps you, I'm assuming that the first one you put there is going to be interface. It's just kind of default assumption. If it helps you to memorize it, that's certainly fine to do it. As a matter of fact, that makes a lot of sense that you do it that way.
Okay.
To drill it. You guys might want to, maybe one of you guys share your screen so that people can kind of see. That might help. Right.
I don't know if my Wi-Fi could handle that. It's been going off and on all day, so it's
Yeah, well, yeah, let somebody else do it. You can kill your screen too if you need to. Which one do you think is first? I agree. I was gonna say, drop it and then you click it, press click, and drop it over there. You gotta cut it. Whoop. And that's- Which one do you think is first? So, and then right click, and then you cut. And drop it right. See, in the first one. Nice. Oh, you're.
Do we delete the first image?
Yeah, you, I mean, it doesn't matter. Yeah, if you copy it, you can do it. You can cut it too. What are you trying to say, Colin? Well, you have a the cord for it? I'm running extension cord from back there.
Well, we have to leave the other side empty.
I don't care. It sucks to do it with, that way you don't have to sit there and struggle. It's taking a minute from the load. So which image do you think is the first one? Which do you think over here is the first? That one? I agree. That's interface. You click it and then right click. And you drag it in the next. You can right click now. So once you click on the right click. Forward. doing that first one. Okay. It should give you the option to cut. I don't mind doing that. I don't know how to do that. Okay. No, I want to do it. Can I touch it? No. Let me see what happens. Okay, so it's probably going to be five. I would do control C, see if that'll let you stop it. And then control V, if you pay. See if you can't do it. Just come back up here and you control V, see if it'll let you pay. All right, yeah, that's what you have to do. So you gotta get all these over here in the correct order over here, and then you gotta do everything. Or why you put them in another. What is that? Hands. Y'all stay, don't leave your group until it forces you. Y'all coming back soon, man. Y'all still had time. You still have 1 minute.
This is due by midnight, right?
Yep. Force you to think about it. Let's see.
I feel
This
like
is-
this,
You feel like what? You feel like what again?
this New Year was probably the most celebrated one in a while.
Probably. I was asleep at midnight. I don't know. I don't know about New Year's. Okay, cool. Everybody's back now. Excellent. So there is a document, and I'm going to work our way through it, and I'm going to have you guys help me work our way through this. So I'm going to expand this so that you can see everything, and I'm going to need to write on it. I might actually just download it. Might be easier for me just to download it. Use the document that way. Do I need to reshare my screen, or can you guys see the Word document open?
We can see it.
All right, cool. All right, I'm gonna ask for some help from some of you guys, but I want to make sure that we got plenty of time to practice. And we only have 5 minutes before I have to let these guys in here get ready to leave for their next class and clean their desk. So I plan 65 minutes. All right, so I'm going to use the annotate button, and I will do the very first one. So it says the micrograph. So this micrograph here, a micrograph is essentially a computer generated image of, yes ma'am. Say that again? Oh, crud. Oh, there's several people who got kicked out. Sorry, hold on a second. Let me let these kids in. Several people got kicked out. Sorry. All right, so you guys are back. Okay. So a micrograph is just a really a picture of taken from a microscope of some cells. So I've got 4 cells. Which one is actually in anaphase? So you got A, B, C, and D. Which of these do you guys think is anaphase? So let me ask Tarleton. I haven't spoken to Tarleton today. Tracy, which do you think?
Oh, could you scroll down a bit?
Yep. Which of these images do you think is anaphase?
Are you marking up a Google Doc?
Nope, I'm marking up.
We can't see it.
Oh,
Word
y'all can't see
document.
it. Oh, yeah, that's what I was asking. Can you
Yeah.
guys see that? Oh, that helps. Can you see it now?
Yeah.
All right, cool. So which one, Tracy? I've got A, B, C, and D. I'm sorry?
D.
Yeah, you can see it. That's going to be anaphase for sure. You can see that there's probably a centriole here, a centriole here. I've got my sister chromatids on being pulled apart, being pulled away from each other. That's definitely going to be anaphase, the away phase. Not telophase. If it's telophase, they would already be separated. Maybe these had just went through cytokinesis, but they're definitely not going to be telophase. Okay, then that's it. So let's go to the next one. The next one, do you need a calculator? You don't actually need a calculator. You can do it without a calculator. Let me clear this image here. Come on, clear. Clear all drawings. Now, let's see. So it says, what is the meiotic index? And you guys probably don't know what a meiotic index is. A meiotic index is one that wants to know what proportion of the cells are in mitosis versus interphase. So what you have to do is you have to get the total number of cells. So T is going to equal total number of cells. There are 10, let's count them. There's 10, 13, 15, 5 is 20, 20 plus 30 is going to be 50. So I've got 50 total cells, 50 total cells. How many of these cells are in mitosis? Well, prophase is part of mitosis, metaphase is part of mitosis, anaphase is part of mitosis, and telophase is part of mitosis. So 10 plus 3 plus 2 is 15 plus 5, that's 20. So 20 divided by 50. So this really is metaphase. So it's metaphase by total number of cells. Metaphase by total number of cells. So what is 20 divided by 50? I'll ask one of you. Brandon, 20 divided by 50. What you got? What's 20 divided by 50, yo? Yep, 0.4. So if I can get this sucker to draw, 0.4 is correct. So there's my answer. So meiotic index, I just want to know what proportion. That's a pretty high meiotic index, to be honest with you. So this would probably be an organ that's rapidly dividing, All right, I'm going to stop here. We will start on number 3 to as a do now in the next class, and then we're going to migrate. I want to give you guys more time. Today's a little shorter than normal. At the bottom of the document that you guys were working on, there are 3 questions. You need to answer those 3 questions successfully, and that is your homework. Just that document that we had 15 minutes to work on, and I won't grade that until, I probably really won't grade it until tomorrow morning. So if you guys have it before, you know, literally before class, it won't be counted later on. I will see you guys in the funny pages. I gotta let these guys clean their desk. All right, adios, kids. All right, so you guys, you gotta, all right, bye-bye, be good. So I'm gonna come around